Hi, and welcome to this brief tutorial on how to understand what the Rh factor is in blood typing, as well as how to do a genetics problem involving this blood group. My name is Dr. Randy Papke, and welcome to this tutorial. The first thing that I want you to know is that the Rh factor is a blood group very similar to the ABO blood group that most of us are familiar with. The Rh factor is named Rh because we share this protein that's found on the outside of our red blood cells with rhesus monkeys. In fact, that's part of the reason that this blood type was discovered was because of the compatibility with rhesus monkey blood. So what does it mean when you're type A positive or type O negative? The letter A or B or A, B or O, that's a separate blood group. So when you say A positive, you're actually talking about two separate blood groups, the ABO blood group and the Rh factor. And because the Rh factor is kind of a simple dominance and recessiveness, we basically just say whether you have it, that means you're positive, or whether you don't have it, that means you're negative. If you have the Rh factor, then you have an antigen on the outside of your red blood cells known as the D antigen. There are other antigens on the outside of your red blood cells, but we're going to ignore those for now and just focus on the D antigen. So again, if you're Rh positive, you are able to make this factor, this protein that is on the outside of your red blood cells. If you're Rh negative, then you are not able to make this antigen, and your red blood cells look kind of naked. Like they don't have this particular protein on the outside of the red blood cell. So because it's either present or it's absent, it's present in Rh positive, the D antigen is absent in Rh negative, we have a very, very simple dominance and recessiveness situation going on here. So let's take a look at a genetics problem looking only at the Rh factor. So the first thing that we're going to do is present you with a problem. Here's the word problem. You've probably seen things like this in genetics problems before. What is the predicted phenotypic ratio of a cross between a woman that is heterozygous for the Rh factor and a man who is Rh negative? Whenever you're presented with genetics problems, it's a really good idea to first write out the alleles associated with that problem. Remember before I told you that the name of the antigen is the D antigen. So we're going to use the letter D to represent the alleles for the dominant having the Rh factor and the little d as the recessive being Rh negative not having that Rh factor. So we've got that down. Now all we have to do is kind of pull apart the sentence. We've got a woman that's heterozygous and a man who's Rh negative. Heterozygous means having one of each allele, and the man's Rh negative, so he must have two of the little d alleles, because that's the only way that you would be able to not have any of those antigens on the outside of your red blood cell. So here's the cross. The woman is big D little d, she's heterozygous, and the man is little d, he is homozygous recessive. If you watched any of my videos before, I like to then be really explicit about what happens next. And what happens next is that we go through meiosis. So let's take a look at what happens if the man and the woman go through meiosis on their own. Women make eggs and men make sperm. So we're going to write those gametes down below the cross in something that I call the table of gametes. And this is what you would draw, like a little T shape for a table underneath of that cross. And then you could fill in the woman's gametes. Remember, gametes are haploid, so we would separate the two alleles into separate cells. And then this is what the gametes for the man would look like. Once you've made the gametes, you can put them into a Punnett square. And most people who have done genetics problems are familiar with the Punnett square. That's our next step. And this is what a Punnett square would look like. So all we do is take the gametes that we made earlier and we put them on the outside of the Punnett square. You can see here that the woman's gametes are on the left-hand side of the Punnett square and the men's gametes are going across the top of the Punnett square. Now we simply fill in what would happen if one of those sperm met up with one of those eggs. And in the first box, it would be a heterozygous offspring. And that's true for the second box as well across the top. But if we look at the bottom left-hand corner, that's a little d sperm meeting up with a little d egg, and so we have a homozygous recessive offspring. And same for the final box. So we've predicted our offspring. And step 5 in doing a genetics problem is always make sure you answer the question. And so doing the Punnett square isn't enough. You need to answer the question. And the question says, what is the predicted phenotypic ratio of the cross? So we need to write that down. And remember that our phenotypes were Rh positive and Rh negative, which look a little different from our alleles. So in this case, we would say in a simplified form, we have one Rh positive to one Rh negative offspring. You could write this other ways. You could write it as 2 Rh positives to 2 Rh negatives, because that's how many boxes you have. You could say 50-50, you could say 1/2 to 1/2. All of those would be accurate ways to answer the question of what is the predicted phenotypic ratio of the cross. So that's good, understanding a little bit of how to do the genetics. But let's bring back that ABO blood group and see what it would look like if you had individuals where you were thinking about both ABO and Rh. Here you see an individual that's AB positive. They have the A antigen, the B antigen, and the Rh factor antigen. If you're A positive, you have the A antigen and the Rh antigen. If you're B positive, you have the B antigen and the Rh antigen. And if you're O positive, you have neither the A or B antigen. That gives you the O factor. Like O is like nothing. It's like naked, kind of like Rh negative. But in this case, it's O positive, so we do have the Rh antigen. If we look at the negative line here, we see that there's no Rh factor on any of these individuals. And if we look at the O negative blood group right here, the blood type right here, you can see that they have neither the A nor B antigens from the ABO blood group, nor do they have the Rh antigen from the Rh blood group. I hope that was informative and helping you do your genetics problems in your biology class.